Today it's my pleasure to talk with David Datta of Harvard University, where he's professor of neurobiology and an investigator at the Center.
My name's Bob.
Oh, what did I say?
You're thinking David. You're thinking of Ginty.
Oh, I was thinking of David Ginty.
We should probably do that again. I'm okay with anything, which my name should probably be right. That's all.
Yeah, Bob and I were talking about his department chair before we went online. And so he's at the Center for Brain Science at Harvard, which is I guess, interdepartmental collection of scientists working on all aspects of brain science. And the overall goal of research in Bob's lab is, and I'm taking this from his lab page, how the brain sees the outside olfactory world. And it goes a little bit beyond that, involving and we'll talk about, I'll have him talk about this, something called computational neuroethology, or kind of quantitative. How do you actually quantify natural behaviors of animals in their environment? Because most of the work on behavior in laboratory animals is in very unnatural settings. And so maybe at the end, after he talks about the olfactory system, we can talk about that. So you're an MD, PhD. Why don't you start there and talk about your journey to where you are now as a full professor at Harvard?
Sure, yeah. So I was an undergraduate at Yale, where I studied fly development. I was fascinated by how, from a genetic blueprint, complicated animals can emerge. And so, at the time I was studying as an undergraduate, that was kind of the heyday of people thinking about the body plan and how homeodomain proteins, lead to the establishment of various axes and segments. And so I really cut my teeth thinking about gene expression and development. I then came to Harvard to do an MD PhD, and I kind of continued along that line of work. I spent a lot of time kind of rotating through labs thinking about gene expression, and I came across this guy, Mike Greenberg, who was studying gene expression but in the nervous system, and that seemed like this incredible opportunity and challenge. And just a few years prior, Mike had developed basic technologies that were amazing then, kind of still are amazing now, but we take for granted now, that allow you to culture neurons and introduce foreign genes into those neurons. Right? That was like an unheard of thing, because those kinds of experiments allow you to think about causality. Right? You can put in dominant negative molecules or molecules that have gains of function, and think about how individual neural cells functioned. And this fascinated me. And so I joined his lab for my PhD, which took 7 long years, during which I focused on studying how signal transduction pathways support the survival of individual neurons. So it's kind of well known that growth factors during development prevent the death of cells. And my thesis was really about working out why that is at a very granular molecular level. When I was kind of finishing up my PhD, I realized that I was probably not going to practice medicine. I loved seeing patients and the experience of being in the hospital. But at the same time, I knew that I just couldn't live away from the bench. And so when I was a 4th-year medical student, I took 6 months off to do a research rotation with my friend and colleague, and now my next door neighbor here at Harvard, Bernardo Sabatini. And at the time, Bernardo didn't really know that much molecular biology. And I knew nothing about systems neurobiology or functional imaging or anything like that. And so we basically arranged this deal where, I would teach his people how to clone genes, and he would teach me how to build a 2-photon microscope. And for both of us, that ended up being like this really important and informative experience. And we still collaborate to this day. But it really changed the trajectory of my work. And it made me really want to, in many ways, kind of combine molecular neurobiology with a kind of systems-y perspective. Yeah. So then I went and I did a postdoctoral fellowship with Richard Axel at Columbia, where I studied olfaction. And in particular, I studied neural circuits that support the generation of behaviors in response to pheromones. So I developed some new neural tracing techniques, based upon the multi-photon experience I'd received in Bernardo's lab, that allowed me to kind of reach into the fly brain and to understand why behavior that's elicited by the same pheromone in a male fly is different from the behavior that's elicited by that very same signal in a female fly. And at the end of that experience, I decided to switch my lab back to mammals. So I came here to Harvard as an assistant professor and decided to work on the mammalian olfactory system. And that and the thought was at the time that I would try to understand how odors elicited innate behaviors. And in this case, the innate behaviors we're thinking of were pretty simple. Things like, why does a mouse run away from fox odor? Why does a male mouse love to sniff female mouse urine? The kind of very simple, basic innate behaviors. And we realized kind of in doing that actually we have no idea what mouse behavior actually is in any real sense, even in the baseline. So like, what is the time varying structure of mouse behavior as it runs around in a bucket? Because if you don't understand that, you can't understand how things have changed when you give an odor. So my lab kind of, I wouldn't say quite bifurcated, but kind of ended up focusing on two, to us, very related, but somewhat separate topics. One, we think about how mice use olfaction to navigate the world, right? Smell is the most important sense for most animals, certainly including mice. And so we think a lot about how information about odors is encoded and detected in the brain, passed on to higher olfactory centers, processed. And then we also think about the other end of it, which is, how is natural behavior built, in mice? How do mice, shape their behavioral dynamics over time during, in naturalistic settings and doing kind of naturalistic things. And the long-term vision for the lab is really to unite these two parts of the lab, to think about how, mice use their sense of smell to better understand the world around them through behavior. And so we've been slowly but surely building up kind of tools and technologies and concepts that are going to hopefully one day allow us to really think seriously about how mice take advantage of their sense of smell and their ability to shape their own behaviors to build maps of the world that are useful.
And then you mentioned, odors from predators and odors from the opposite sex. Mm-hmm. A mouse as being kind of innate, evolutionarily critical for survival and reproduction, right?
That's right.
But when a mouse is, out in the woods, I'm at home now and we've got woods behind us and there's undoubtedly mice out there. There's definitely squirrels. And so they're moving around in the woods and there's a lot of, and their sense of smell, they're more sensitive than us, just like, I think, right? Like,
yeah, I think we have, there's a lot of debate about this because I think people have long thought that the human sense of smell has been underestimated, but by the numbers, it's a sensible inference that mice are better smellers than humans. So just to give you one example of that, the olfactory receptor genes are the largest gene family in mammals. So more space in your genome is devoted to smell than to any other kind of biological function. In your genome, there are about 400 receptors for smell. In a mouse's genome, there are about 1,000. So, and if you look at the kind of amount of neural real estate devoted to smell, in the mouse, it's about 1/3. Whereas in primates like me and you, about 1/3 of your brain is devoted to vision, which makes sense, right? So just in terms of the numbers, it seems pretty reasonable to think that mice are better smellers than we are.
Okay, so talk about the olfactory receptors. Obviously, they're in the nose, mm-hmm. and you said, say in mice, there's up to 1,000 different receptors.
That's right.
So they're on the surface of. Does each cell have only one type of receptor, or do they have multiple?
Yeah. So your nose and my nose and the nose of all vertebrates is kind of coated with a sensory epithelium, right? Which makes sense. Like all sensory modalities work through these kinds of epithelial sensory sheets. And the same is true for smell. And this olfactory sensory epithelium is filled with olfactory sensory neurons. So this is a sheet that lies outside of your brain that's filled with primary sensory neurons. And these neurons elaborate little cilia that point out towards the airspace and are coated in mucus. And the very tips of these cilia are really jam-packed with protein for odor receptors, which are 7-transmembrane G protein coupled receptors. And it turns out that in mature olfactory sensory neurons, like once the neuron is made and mature and integrated into the circuit, they only express one of the 1,000 possible types in the mouse. This is the so-called one receptor per neuron rule. And that rule kind of governs the overall logic of how the system works. We don't fully understand molecularly how this singularity is achieved. It's a really interesting problem. I mean, the other example where you have this kind of genetic complexity is the immune system. It's clear that the mechanisms that ensure that each olfactory sensory neuron expresses only one receptor are different from the mechanisms that, for example, allow a B cell or a T cell to only express one B cell receptor or one T cell receptor.
Huh.
So there's something very unique going on in terms of molecular mechanism. But regardless of how it works, it's pretty clear that each neuron expresses only one of these receptors, and that receptor, the expressed receptor, determines the tuning properties of that cell. And so you can think about these odor receptors, each of which has a kind of specific set of odor tuning properties, right? They interact with different odors in the world, but the tuning properties are actually pretty broad. So each receptor seems to interact with a fairly broad set of odorants, although each receptor is dedicated to a kind of unique part of odor space. And that kind of helps to explain why we can smell so many things. So like we only have 1,000 receptors, but it's not like you smell an odor and that odor only activates one receptor type. Typically, in our data and data from others, a typical environmental odor will activate 10 to 30% of the receptors.
Oh, huh.
And so the way that you understand, what odor you're sensing is really, based upon which combination of receptors is activated, rather than the, activation, of a single specialist receptor in general.
And are these receptors coupled pretty directly to ion channels then?
Yeah, so it's funny you asked that. So it turns out that there is a well-defined signal transduction pathway that runs from these G proteins through specific adenylyl cyclases and then adenylyl cyclase dependent ion channels to generate spikes.
They're all cyclic AMP.
That's right. Actually, part of the way, yeah, exactly. They're all cyclic AMP dependent. That's
Okay.
right. And one of the interesting things that my lab has recently discovered is that we pretty much know the set of genes that connects the GPCRs that detect odors to the generation of spikes. And they include, cyclic AMP metabolic enzymes, right? Ion channels that respond to cyclic AMP, phosphodiesterases, calcium binding proteins. And it turns out that there's a really interesting molecular program that coordinates the expression of all of these genes and kind of turns them all up or down adaptively. So if you're in an environment where a particular olfactory sensory neuron that's expressing a particular receptor is being subjected to a lot of activity, transcriptionally, that cell will turn down the expression of all 70 of these genes, make this coordinated rheostat-like fashion. And conversely, if like a neuron isn't particularly active, that program gets kind of progressively turned up, kind of adjusting each neuron's sensitivity to odors depending on what's in the world around you.
And we've all experienced that. For example, what popped into my brain was being around someone who smokes, right? And like even their clothes, the odorants in their clothes, they may come into room. That's just one example of many types of that adaptation.
That's right. That's exactly right. So in olfaction, there are many, like as in every sensory system, there are a lot of different mechanisms for adaptation. But in most systems, including in olfaction, people especially for longer term olfaction, longer term adaptation over minutes or hours. People typically think about circuit level mechanisms, like your brain turns itself down. And what's interesting here is that through transcription, the neurons turn themselves up or down, on timescales that are transcriptional, so minutes to hours.
Yeah.
And our thought is that, this kind of adaptive mechanism helps us as humans and most animals kind of adapt to the environments we are soaking ourselves in chronically during the day. We're in one environment, we're at home, then all of us come to work, we're in a different environment, then we go back home. And, our noses probably use this kind of transcriptional adaptation to adjust, on long time scales to environments we're in for a while.
Yeah. Okay, and then, so the odorant comes into the nose, it activates multiple cells. Each cell has only one type of receptor, but a given odorant binds to more than one receptor, and then somehow that information gets up to the brain. What is that?
Yeah, right. Yeah. So believe it or not, we know a lot about that, but nowhere near enough. So it's clear that, there are these neurons that are in your nose, and their axons traverse a bone at the base of your skull called the cribriform plate, which is filled with these little tiny holes. And those axons then terminate in a structure in the basal forebrain called the olfactory bulb. And that weird anatomy where you have a bunch of neurons outside the skull, and then their axons puncture the skull to talk to the brain. That weird anatomy explains why, if you're a boxer and you get hit hard in the nose, you lose your sense of smell, right? Because the cribriform plate will displace or crack when you get hit in the nose, and that shears the axons. They're poking through that plate, actually. And so a very common way to be rendered anosmic, before COVID, was to be in a motorcycle accident and fall, hit your nose on the pavement, or to be a boxer, and then to lose your sense of smell through trauma, and shearing of those axons as they enter the brain.
Before you move ahead, since you mentioned COVID, I see recently you're on several papers looking at, and that's one symptom in many cases, and you had some interesting findings concerning which cells, I guess in this nasal epithelium.
Yeah, that's right. That's right. That's right. So, when the pandemic started, I think, the initial surprising thing about it was that everyone who got COVID lost their sense of smell. And that's super weird. Like, even if you get, if you or I have a rhinovirus, just get a cold or something, we lose our sense of smell some of the time, but not all of the time. And nearly always, the reason why we lose our sense of smell is largely because our noses are filled with mucus. Like, it's this an inflammatory response. There's this kind of obstruction of your ability to smell things. In a day or two, you take some Nasonex or whatever, and your nose clears up and you can smell again, right? That's the normal course of events. And actually, because it's so benign, we actually don't know much about how viruses attack the sense of smell. And then all of a sudden, COVID comes along, March of 2020, and everyone's actually losing their sense of smell. And they're losing it, for a week, 2 weeks, 3 weeks, but some people for much longer, months or years. The sense of smell kind of is lost in an instant. So it's not really associated with inflammation. Many people are breathing normally. They can get air up and down their nose. There's not a lot of mucus, but they just can't smell at all. And so we knew that something really weird was going on. And the other weird thing about it was, and I think this is if you think about it mechanistically, it's really surprising, is that basically it was kind of like a light switch was getting turned off. So you could smell, then all of a sudden you couldn't smell. And then a week later, 2 weeks later, it's like the switch was just flipped back, and all of a sudden you can smell again. And so, it was really mysterious how that might work, right? So the intuition would be, if you didn't know anything, that the virus would be attacking the neurons responsible for detecting odors, right? But what you kind of know that can't really be true. And the reason you know that is if the virus is getting into your nose and killing the neurons that are responsible for detecting smell, your nasal epithelium has stem cells that can rebuild and continuously during your normal lifespan, actually rebuild these neurons. So unlike, most neural populations, you have stem cells in your nose that are constantly spitting out these olfactory sensory neurons and integrating them into the circuit. So these new neurons are capable of being built and then elaborating new axons and connecting to your brain, right? And that happens all the time during your normal lifespan. And it's known that when you're infected by a virus, if some neurons die, you can replace neurons through this mechanism. But the problem is that, it takes months for that process to work, right? So that your epithelium doesn't turn over in days, it turns over in weeks to months, right? So that already makes it seem like killing neurons is not the mechanism through which you're losing your smell and then getting it back. Something else is going on. Mm-hmm. So me and a bunch of colleagues of mine, it's kind of scattered across the globe, for a whole variety of reasons, had been doing single cell sequencing of the nose. And so we have, a kind of giant matrix with cell types on one axis and all the genes on another axis. And so we could kind of sort through all the different cell types and ask a priori which cells are infectable by COVID, by SARS-CoV-2, which is the virus that causes COVID. And of course, it's known that the major receptor for SARS-CoV-2 is this protein called ACE2. Yeah. So a simple thing to do to kind of figure things out is just to ask, which cells in your nose actually express ACE2. And it turns out that the sensory neurons do not. They don't express ACE2. Mm-hmm. But these sensory neurons are actually interdigitated in the olfactory epithelium with these support cells, which are called sustentacular cells. Sustentacular for sustain, right? And these cells kind of prop up the olfactory sensory neurons, maintain appropriate ion balance so that the sensory neurons can fire, and actually are thought to even be involved maybe in the metabolism of sensory neurons, or thought to, like feed energetic intermediates, the sensory neurons, to help support them.
Like lactate or
pyruvate. Yeah, exactly. Even ATP, I think. And although these support cells are super mysterious, and actually there's very little work on them up until this point. But it turns out those sustentacular cells express this ACE2 protein, the receptor, COVID gangbusters. And so we kind of hypothesized early on, based on these data, that the mechanism through which you lose your sense of smell is that these sustentacular cells are getting infected. And that explains why, like, you can have an infection that kind of causes you to suddenly lose your sense of smell. Maybe the metabolic support goes away when these sustentacular cells are infected. And then when you clear the infection and your sustentacular cells recover, you still have the neurons intact, right? Yeah. And so then you can just regain your sense of smell. And so that's the working model we articulated back in 2020. And evidence from animal models and now from humans, including work that my lab has recently done with our collaborator, Brad Goldstein at Duke, is consistent with that hypothesis. So if you look in autopsy patients that have died, unfortunately, from COVID, you don't really see neurons in the nose infected, but you see plenty of sustentacular cells infected. We did a recent study, which we think is really interesting, in which Brad, who's an ENT, he's a surgeon, actually did biopsies of patients that had recovered from COVID, but objectively still have lost their sense of smell months later. And so we can grab
nasal epithelial biopsy.
Yeah, exactly. So it's actually, if you think about it for a second, it's this kind of remarkable scientific opportunity. So it's a chance for us as basic scientists to get our hands in a living patient on nervous tissue.
Yeah.
Okay. And so he was able to do these biopsies, and we don't see that the neurons are infected in that setting either. And what it looks like in patients that kind of still can't smell months later is that there seems to be some sort of immune reaction, such that your epithelium is kind of filled with monocytes and macrophages that are causing a lot of local immune signaling. And it seems likely that local immune signaling is suppressing the ability of your sensory neurons to work, which is interesting, right? It suggests a strategy for maybe helping people who've lost their sense of smell.
Now, so SARS-CoV-2, it won't infect mice, will it?
That's right. So, and actually a lot of the confusion about how SARS-CoV-2 affects smell comes from the fact that it doesn't really infect mice. That's correct. And so what people have done is they've genetically engineered mice to express ACE2 in all neurons. And if you take a mouse in which ACE2 is expressed everywhere and you give it COVID, then all the neurons that are ectopically expressing ACE2 get infected. And so you might think erroneously that means that in patients, that's what's happening. And that's, of course, not what's happening. But at the beginning of the pandemic, there was a lot of confusion because mice don't really get infected. So believe it or not, the The best rodent model we have is the golden hamster, which actually gets infected quite readily, and it's been used to model the evolution of SARS-CoV-2 infection, both in the lung and in the nose. Where
that's interesting that hamsters seem to be, we did a lot of work on amyloid precursor protein and amyloid beta peptide, and the sequence, APP sequence in hamster and A beta, amyloid beta peptide, is the same as in humans.
Yeah.
But it's different than mice.
Yep.
But the experiment I was thinking of, okay, so ACE2 is in these sustentacular cells, and then, if the mice were normally infected and they had loss of smell, then you knock out selectively ACE2,
right,
and then see if they don't lose their smell anymore.
Right, exactly. That, as far as I know, that experiment has not been done in the hamster. What has been done is to look at like the dynamics of what happens after you infect a hamster epithelium in an intact hamster with the virus.
Yeah.
And what you see is that the sustentacular cells get infected, the neurons don't. But if you really infect the tissue, substantially, then the sustentacular cells themselves eventually die, and they take the neurons with them. And so we think that's like a pretty viable model for why some patients lose their sense of smell for a long time, and it takes a while for them to recover. It's because like they had a really strong initial infection that actually knocked out the tissue, then the whole tissue has to rebuild. And that, as I mentioned before, takes weeks to months.
Yeah. Okay, so I'll get back to.
I want to say one last thing about this, because like we're doing a really cool experiment, and it's like the coolest experiment. I just want to like talk about it because it's cool.
Okay.
Which comes out of this line of work. So, one of the interesting things that is a consequence of people losing their sense of smell with COVID is they seem to be like more likely to agree to a nasal biopsy. Like people are just like curious about how smell works now. And so like if you just like ask people on the street, hey, can I take a punch biopsy of your nose for science? They say yes to that, which is amazing.
Well, but that makes sense. They have a vested interest. When I was at University of Kentucky, they had actually really good autopsy program for Alzheimer's disease.
Yeah.
And the thing in that area is it's hard to get autopsy material brains from neurologically normal subjects.
Right.
So they, talk with the spouse of the person. And, so anyway, they had this autopsy program where they'd recruit significant others of
makes sense, right? Yeah, makes sense. Makes sense. Yeah. So we're taking advantage of this fact to do some really interesting basic science. So it turns out that if you expose a mouse to a smell, okay, so you imagine you have a mouse and you expose it to a smell, and I told you that in a typical mouse, 10 to 30% of the neurons are going to be activated by a smell. Okay. So what you can do is, instead of like assessing the activation of the neurons through physiological measures, you can actually do single cell sequencing right after you give the mouse the odor. And then you can identify all the olfactory sensory neurons that were activated through the expression of new genes, immediate early genes like FOS and other genes, right? Yeah. And because it's single cell sequencing, you can also identify the receptor that's being expressed, that's responsible ultimately for the induction of those genes. And so now you have a kind of platform in vivo for taking any odor and understanding and identifying all the receptors that respond to that odor. So now you can finally kind of solve this basic problem in physiology, right? Which is like, what is the mapping between odors and receptors? And Brad and I realized that we could actually do this in people. So we've begun doing experiments where we give humans smells, and then we punch biopsy their noses, do single cell sequencing, and try to clone the human receptors for odors. And we have done a proof of concept experiment. It worked the first time. We are super excited to go into humans. And like, and what's interesting there, actually, like if you just think expansively, these are real human neurons, right, that are actually responding to a physiological stimulus. So you could imagine doing this not just in normal controls, right, but in people with a whole variety of neurological diseases, to think about whether or not we can now get a kind of functional readout at the molecular level for the way that neurons work in a particular person, right? And so we're feeling excited and ambitious about this as a way of interrogating the function of the human nervous system.
So when you do the biopsy and take the olfactory epithelium, you're doing acute experiments. Can these be maintained for days and weeks in culture?
Yeah, I think there are experts who can do that. We typically just grab the cells and immediately sequence them to understand what responded and what didn't.
Yeah. Okay. All right, and then move on up to the olfactory bulb and
yeah. So then so it turns out, just to kind of give you a sense of it, that I don't know, in the mouse nose, there's like 20 million olfactory sensory neurons, something like that. Okay, so millions of neurons. And I told you that each neuron expresses one receptor, right, of 1,000. And so what that means is that on average, a mouse nose will contain 10 to 50,000 neurons expressing a particular receptor. Okay. All of the neurons that express the same receptor send their axons to the olfactory bulb, and all those axons converge on a single one or a handful of insular structures called glomeruli. So if you imagine you have 1,000 different populations of neuron in your nose, that means there's 1,000 different types of glomeruli in your olfactory bulb. And what's amazing is that the spatial location of each glomerulus is stereotyped across animals. So there's this really complicated axon guidance problem where, when you're generating the system, each olfactory sensory neuron, based upon what receptor it expresses, has to send its axon to target the correct one out of 1,000 of these things. And actually, one thing that people with COVID are complaining about is something called parosmias, where smells smell funny. Like you smell something and you expect it to smell like lemon, but it smells like gasoline. And we don't really understand why that's happening. But one possibility is that when the epithelium rebuilds itself and these new neurons are sending their axons up to the cribriform plate, they're actually mistargeting and going to the wrong glomerulus initially, which eventually gets refined and corrected. But because of the initial imprecision, you misunderstand one smell for another. That's the current model for that. So there's this incredibly precise and still not fully understood process through which these 1,000 different information channels in your nose are organized into this kind of crystalline spatial array in the olfactory bulb, right?
Right. So now there's kind of a map already.
That's right.
Spatial map of the odor environment.
That's right. There's a spatial map of the odor environment that lives in your olfactory bulb. I think current research suggests that different odors both activate different glomeruli, and the timing with which those glomeruli are recruited might vary depending on the odor and whether the odor is binding strongly or weakly to a particular receptor. So we're now used to thinking about the olfactory bulb as containing kind of a spatiotemporal map of the odor world as you kind of are smelling things.
Yeah.
And then there's some processing that goes on in the bulb, and then that information is sent to a bunch of different regions in parallel that somehow collectively collaborate to support odor perception. And what's neat about olfaction is these areas are areas that for the other senses take a bunch of synapses to get to. So for example, so obviously information goes from the olfactory bulb to the olfactory cortex. That makes some sense, right? And we think our general understanding is that the olfactory cortex is important for odor discrimination and for odor generalization, for helping us know that lemon and orange are different, and also for helping us know that lemon and orange are both citrus, right? Yeah. But information also goes directly from the olfactory bulb to the hippocampal formation, to the entorhinal cortex.
That makes sense.
And it's thought that the intimate connection between your nose to the bulb to the entorhinal cortex is part of why smells evoke such powerful memories. It's also true that information goes from the olfactory bulb directly to the cortical amygdala, which directly feeds into the amygdala. And it's thought that that's possibly why smells evoke such profound emotions in people. Yeah. And of course, this is very different from all the other senses, which go through the thalamus and then have to hop a bunch of synapses before they get to hippocampus or they get to amygdala. So there's something really primal about olfaction, from an evolutionary perspective, such that these very ancient systems for navigation and emotion and memory are like one synapse away from the outside world.
Yeah.
For smell, but not for your other senses.
And you mentioned the hippocampus and memory, in this case, remembering odors, but the hippocampus is very important for spatial navigation.
That's right.
And obviously, that's one thing that odors are doing, right?
Yeah, right.
The animal is, what odor, what's the relative intensity, I guess, or concentration in the air?
Yeah.
and they can kind of use this information, help them navigate towards or away from,
Right.
even from, from objects they don't see. That's why it's
Right. Yeah.
so important. Why it's
Yeah,
so
that's right.
important.
Yeah, and actually, so it's obviously true, right, that for animals that aren't us, that aren't primates, the sense of smell is really important for interacting with the world at a distance. When like animals crawled out of the muck and became terrestrial, they largely were nocturnal. So like they don't have vision. They're dealing with the world around them initially, basically through their sense of smell, right? And obviously we have eyeballs that can saccade. So, for us, we can interact with the world at a distance in a different way. But for most animals, smell is really, really, really important for kind of allowing them to situate themselves in the world. And I think one of the main complaints of people who suddenly lose their sense of smell is they feel a little bit emotionally adrift. And part of that is that it's probably true that even in humans, the constant, even subconscious stimulation we get from our noses helps maintain a kind of emotional tone that helps us feel centered in the world.
Yeah.
And it's also probably true that there are ambient odors that help orient us in space that we're not even aware of. And if you lose those cues, you feel kind of lost. And so, yeah, so I think that smell, in lower animals and even possibly in us plays an important role in navigation and in understanding the world around us from a spatial perspective.
Yeah. I guess humans figured out, even though they had no idea how it works, that, for example, if I'm talking about pre-agricultural revolution type, hunter-gatherer type situations, right? They learned if the wind is blowing this way, you want to be downwind of it so that
that's right. So
what you're hunting.
Yeah, exactly.
Yeah.
That's right.
Okay, so then you mentioned the olfactory cortex and the cortical amygdala, which amygdala is involved in emotion, fear response. It's been most studied for, and but then somehow there has to be, that's kind of the sensory side of it. But what about the response? How does odors then through cortex, somehow it has to signal to motor cells?
That's right. So maybe it surprises you, maybe it doesn't. No one knows. So actually, it's clear that odors are capable of evoking all sorts of behaviors, and I think to a first approximation, we don't understand anything about how that occurs. It's true that we can kind of trace neural circuits, for example, into the amygdala and identify smell cells, individual cells that are probably responsible for different aspects of fear responses. There's in rodents and in other animals that aren't primates, there's actually an accessory olfactory system called the vomeronasal system that detects pheromones. And it's clear that you can kind of, in a broad sense, trace circuits from this accessory olfactory system into, for example, the hypothalamus, and identify connections that likely mediate various forms of innate behavior. But I think in the details, we don't know much about how smells actually drive meaningful dynamic behaviors. And I think this is actually a really big mystery, and one of the mysteries my lab is very interested in addressing.
Yeah. And so you've studied, used what do you use? Electrophysiology, calcium imaging? How do you look at the activity of, say, neurons in one region of the olfactory cortex when you expose an animal to an odor?
Yeah, so we've done that both by electrophysiology and by multiphoton microscopy. One of the reasons why olfaction there's many reasons why olfaction is less studied than, say, vision or audition. One of the reasons is that as primates were chauvinists, and so we want to study senses that are important to humans, right? But another reason is that it's fairly easy to access under a multiphoton microscope or with an electrode V1. Piriform cortex is at the very bottom of the brain. Getting probes down there, and especially doing direct imaging, has been really a challenge. And so my lab has developed preparations that allow us to do that. And I think there's an increasing number of people who report high density recordings of piriform cortex. But to give you a sense of it, just to get kind of give you a sense, currently, right, there are data sets from Genelia reporting 10,000 simultaneously recorded neurons in V1 in a single experiment. In the pure form, it's probably true that in the literature, in total, there are no more than 2,000 units that have been reported ever in history. Like that's the total number.
Yeah.
And so like our understanding in general is pretty limited just because there haven't been that many total experiments.
And what about, what's the organization in like zebrafish? You know, so you mentioned animals coming out of the muck, but and you also mentioned that these odorants, these chemicals and odors, they have to dissolve in the mucus before they.
Yeah. I know you bring up a really interesting point, so I'll elaborate on it just a little bit. So of course, you know, we all were in the water first, right? And so the original odor receptors were actually evolved to detect soluble odorants. Okay, so in zebrafish, these are the so-called class 1 receptors, and they're really optimized to detect, you know, odors that are effectively dissolved. And so these are the kinds of receptors that zebrafish have, right? And you know, there are all sorts of interesting odors that zebrafish are capable of detecting that are kind of dissolved in the water. Then, you know, we kind of transitioned to land, and we evolved a second class of odor receptor, one that's optimized for identifying and interacting with small hydrophobic molecules that tend to be volatile. So under 400 Daltons, floating in the air, they have to kind of traverse the mucus layer. It's probably true that there are binding proteins that act as chaperones to help these kinds of, you know, volatile odors get across that layer for presentation to the cilia on the sensory neurons, and then you detect them. And what an interesting kind of like side note is that, of course, as terrestrial, you know, land animals, me and you, our class 1 receptors have largely, although not entirely, pseudogenized. So we lose, because, you know, we don't smell underwater, right? So like that whole set of receptors, me and you, and in the mouse, is largely gone. Okay, and so we have, like, you know, residue of that in our genomes, but we mostly have these functional class 2 receptors.
Okay.
So what that means is that if you look at cetaceans like whales and dolphins who were on the land and then went back to water, when they went back to water, they had no class 1 receptors left. And then they pseudogenized their class 2 receptors because now there's nothing in the air because there's no air, right? And so whales and dolphins can't smell, basically. Like, right? Because like they lost both receptor types because they were in the wrong environment, right? It's kind of interesting. So it's thought anyway that for the most part cetaceans are incapable of smell for this evolutionary reason, because of the dynamics of the receptors.
And so, you know, whales and dolphins, as far as we know, I mean, they have bigger brains than a lot of other fish. And so then is their vision better than other fish?
You've reached the limits of my knowledge.
Okay.
I know this factoid to be true, but other than that, I don't know much.
Okay. All right. And all right, so talk about computational neuroethology.
So there's been increasing interest, I would say, over the last 10 to 15 years in using tools from machine vision and from machine learning to find objective ways of understanding the spontaneous behavior that animals generate in the world. I think, you know, everyone agrees that brains evolved to let animals spontaneously generate behaviors of their own free will in response to the world in the way they want to move their own bodies. And most of the ways in which we understand the brain are not in that setting, but instead are in more kind of restricted task-based settings. Often animals are physically restrained or psychologically restrained in various ways and overtrained to express simple behaviors. But, you know, for the most, I think it's not a controversial thing to say that, you know, brains evolved really to allow us to act in the world of our own accord. And so there's been an increasing appreciation, you know, I think by many scientists that coming up with ways to understand the underlying structure of behavior itself, its time varying structure, is going to be important for our understanding of neural circuit function in general. And my lab has developed some of the tools that, you know, folks currently use, and we certainly currently use, to unveil that structure in the mouse. And you know, in the mouse, it turns out that that we were able to discover a kind of hint that has organized our thinking. So I'll tell you what the hint was. So basically, I had this amazing student named Alex Wilchko who decided one day to image mice not in 2D, but in 3D. And the way he did that was to go to the Best Buy, which is just down the street from us here at Harvard, and buy a $90 device called a Microsoft Kinect. So this Microsoft Kinect is a 3D sensor that Microsoft was selling to allow people to play video games. So you put this camera and it would face you and you would kind of dance around in your room, and that would control some avatar playing tennis as part of a gaming console. And Alex realized that he could use this thing, as noisy as it was, to act instead of pointing it forward towards us, point it down towards a mouse in an arena and just watch an animal as it kind of wandered around an empty arena, but watch it in 3D. And so the hint was, if you just look at those data, you just look at them without any modeling or anything sophisticated at all, you just look at the 3D data with your eyes, it's clear that mice generate kind of this staccato type rhythm in terms of their behavior, where they kind of generate a 3D behavioral motif like a rear. They pause for just a tiny little bit, and they generate another behavior like rearing down, and then they generate another behavior like turning right. And about every 300-400 milliseconds or so, on average, they're switching from one little motif to another. And so it's clear, if you just look at the mouse with, in 3D, in the fullness of its pose dynamics, that like the mouse's brain has a kind of underlying rhythm that organizes the mouse's behavior over time. And we were able to take advantage of the fact that this rhythm exists to build telescopes that allow us to characterize and identify all of the behavioral motifs, which we call syllables, that mice exhibit in a particular experiment, to understand how they unfold over time. And one of the exciting things is that we've been recently able to combine this kind of description of behavior, which turns out to actually be very powerful, with neural recording. So now we can stick probes in the brain, and we can ask whether or not we can't come up with neural correlates, for example, for each of these different elements of behavior. But can we identify neural centers that are responsible for placing these behavioral elements in the correct order? What happens if we lesion them, and can animals still generate behavioral sequences? And we've had a lot of fun identifying kind of the mechanisms that at least the rodent brain uses to compose complicated naturalistic behavior out of parts. And we think that a lot of what the mouse brain is doing is invoking these behavioral syllables to allow animals to understand the world around them. And a lot of what the lab's interested in now is asking, how do mice use behavioral syllables or sequences to usefully build maps of the world, maps that are at least in part based on olfaction.
And are you working with like larger cages with a lot of different, almost naturalistic like things? I don't know. Branch.
Yeah.
Rock. Dirt. Is that?
Yeah. So it's interesting, right? It's exactly like the trajectory of this research is exactly as you would imagine. So at the beginning, we have like these kinds of noisy depth cameras, and we can only image mice in empty arenas. And kind of over the last 5 to 10 years, the technologies that support this kind of analysis have improved steadily. And so now we can do many mice in complicated arenas. Just to give you an example, we're working on a paper now where we've performed behavioral imaging essentially from birth till death. And we can look at the behavioral dynamics as they evolve in an individual animal throughout its lifespan. Yeah. And so there's a lot of it turns out there's just a lot of hidden structure to behavioral dynamics that people just haven't had the ability to measure before or identify. And we think this is going to be a very important approach for neuroscience, going forward in the future.
Yeah, a lot of my work over the years had to do with brain aging and neurodegenerative disorders. And so are you seeing, as these animals age, are these the sequencing of these behavioral patterns you're talking about, which I guess you initially did in young animals, is there like a disruption, or are they maintained? Or?
Yeah, it's very interesting, actually. It's the results are much more surprising than I mean, I was surprised and shocked when I myself saw these results. So it turns out that as animals age, there are characteristic changes both in the usage of individual syllables and in their sequencing. These changes are stereotyped enough that we can essentially predict, based on behavior alone, how old an individual mouse is within a couple of weeks. So like it's clear, for example, that the behavior of a 40-week-old mouse is different from that of a 45-week-old mouse. And we can make accurate predictions, of how old the animal is just by looking at it for 20 minutes running around in a bucket. The other interesting thing about that is you can imagine that if you have a cohort of mice that are all of a particular age, so they're all a year old, that you might be able to use the kind of data sets that like I've been describing to build a kind of behavioral clock, right? And so in a cohort of, say, 12-month-old animals, you might see some animals that behave younger and some animals that behave older. That turns out to be true. And we've been able to build pretty accurate death predictors based upon the behavior of mice. So like we can kind of look at a cohort of mice, identify the ones that look like they're behaving like they're younger, look at the ones that are behaving like they're older, and make pretty accurate predictions about, when in the future these mice are going to kick the bucket and in what order, actually. And so there's a lot of, like, I think, we're used to, as humans, thinking of ourselves as being young, middle-aged, and old, but actually it's clear there's this very organized continuum of behavior that's associated with aging that we can think about. And so that kind of project is, underway now, but it's something that we're very excited about.
Yeah, that's really interesting. We did a lot of work on intermittent fasting and caloric restriction too, which I don't know. It's probably not worth you doing the experiments because I think the answers can be predicted that they're going to be beneficial. But what about humans? So you're talking about this high resolution 3D behavior imaging in animals, controlled conditions. Do humans show kind of similar repetitive sequences of just natural, spontaneous behavior?
Absolutely. So there's actually a whole field now of computational psychiatry that seeks to do essentially what we've been trying to do in rodent models. That kind of sensorizes humans, often through smartwatches or other kinds of devices or through video, and parameterizes their behavior and seeks to identify structure. And it's clear that, just the structure of a human with a neuropsychiatric disorder in terms of its his or her behavior is different, right?
Yeah.
And so people are thinking about ways in which they can use these kinds of technologies for diagnosis and for, disease progression and for therapy. One thing I'll say is that the kind of technologies we have been focusing on building are pretty general, and we've been able to apply at least some of them to human data fairly successfully. And so we just think that this kind of way of thinking about biology is pretty general, and it's going to have a lot of yield over the next several years.
Yeah. And I'm almost done with my question, but you use the term pharmacobehavioral space.
Mm-hmm.
Right. And so why don't you just briefly talk a little bit about what you mean by that? And
thank you for asking about that. So, we have this very high resolution way of understanding a mouse's behavior. And so we wondered whether or not we could essentially build a map of drug effects by treating mice with different drugs and then performing this technology that we have developed, which was just called motion sequencing, or MoSeq. So we perform MoSeq analysis on a very large cohort of mice, like 1,000 mice, each one of which we've injected with, one of 30 different drugs at one of, 8 different doses. On the one hand, you can use these kinds of data to benchmark the behavioral analysis technology itself. So you might ask, you might posit that a useful technology would allow you to, through behavior alone, predict the drug and dose the animal was treated with. And that's definitely true. So MoSeq is really great at that. But the more interesting thing is not like, does my machine work? The more interesting thing is, what can I learn from the data, right? And so one thing that it allows us to do is to build drug maps to essentially identify relationships between drugs in terms of their behavioral effects alone, right? So independent of what we know about their mechanism of action, we can ask, net, how are these different drugs related to each other? And that's useful because it in principle allows us to identify drugs that might be very similar to drugs that we like, but have unwanted side effects. So it's a way of nominating candidate drugs that could be useful. And so we've done some proof of concept experiments in this space to identify, for example, candidate drugs that might be useful for treating autism spectrum disorders based upon this kind of pharmacobehavioral mapping that we performed.
So it can be very valuable in, people who are developing drugs in, they need to decide at some point they have drug that affects some target in some whatever disease process, and they have to decide, do we try to move this forward to actually doing clinical trials in humans?
Right.
So this kind of, quantitative detailed analysis you're talking about can help with kind of making early decisions that might in the end make it more likely that a drug that is used in clinical trials actually has desired effect.
I mean, I think the actual, the dream, right, is to use these kinds of quantitative behavioral technologies, both on the preclinical end to identify candidate therapeutics that seem like they're going to have therapeutic effects and translate, but then also actually on humans in the trials. Like you can imagine using behavior to fractionate patients with a given disease into different subcategories that might be better treated by your drug. So you can use these kinds of behavioral technologies to enroll patients in your trial, and which itself might lead to a more successful trial. Yeah. You can imagine these exact same technologies being useful to look for drug effects in patients and look for disease progression and recovery. So we think there's like a very broad application space for this general set of technologies that should help us get drugs into the clinic and into patients.
Okay, so what's your next steps? What's in your grant proposals? No, no, I'm kidding. Okay, but what's kind of your biggest question that most exciting to you right now?
So I think I'm increasingly interested in flipping the problem of behavior on its head a little bit. So, we are building tools that allow us to understand how the brain builds behavior, right? So that's like a big theme of what we're doing. What we have much less of an understanding of is if you watch a mouse running around, say, in a bucket, we don't really understand why the mouse is doing what it's doing. What is the purpose of all this behavioral structure that's clearly selected for by evolution and instantiated by the brain? Like, what do all these little motifs of behavior mean to the animal and to its brain? And so that's where we're heading. I want to use these kinds of technologies to begin to ask about why animals generate the movements they do and what utility those movements have for the animal, right? And that's obviously a very deep question. It's almost a philosophical question.
Yeah, you're getting into the mind of the mouse, and
yeah, that's right. That's right. But like, what are we if not explorers? Let's go try to figure something out, right?
That's right. Absolutely. What's the mouse thinking?
Yeah, that's right. And so we're trying to understand, like, we're trying to build models that explain why, when an animal encounters a new odor, why it moves its body in the way that it does.
Yeah.
And so that's kind of where my head is right now.
And again, going back to the natural environment, right? You're always somehow kind of up, second by second, minute by minute, and then longer term, you're kind of updating your previous experiences,
right?
And then maybe modifying your subsequent.
That's right. That's exactly right. Actually, so like I was mentioning before, the difference between mice and primates, right, is that primates can sit on a branch and move their eyes around to explore the world.
Yeah.
Mice, they have to get out in the world and like physically interact with the world to understand it. And so that suggests that you can use mice to understand the value of movement, and that's the thing that I really care about.
I've often thought that in modern, so there's this evidence that it's just from a few studies that the overall brain size of modern day humans is smaller than before the agricultural revolution, say 12,000 years ago, and that's based on measuring cranial volumes. And so we're becoming very specialized, and you and I, right, or when you get to be a head of a lab, you spend a lot of time doing what we're doing now, right? Sitting at a computer, and we're not moving through our environment while we're thinking. Right. And actually, I find out when I go, and even know this too, probably you take a walk or go on a run.
That's right.
You seem to do a lot of better better thinking than just,
I agree. Like if you're in a circumstance in which you're naturalistically engaging,
yeah,
like your hippocampus, right? Not through necessarily abstract thinking, but through actually moving through the world.
actually moving.
Yeah.
I think,
Something about that's helpful.
I think we
agree,
should be,
yeah.
we should actually be concerned about that, that a lot of people now aren't moving through their environments, and what impact is that having on,
Yeah.
on the brain?
Yeah.
Okay, Bob, thanks. I appreciate you taking the time. I know
My pleasure.
you're very busy, and enjoy yourself as any good scientist should do. Work hard and have fun. That's
Thanks so much. This was a fun conversation. I appreciate it.
all right. Bye.
Take care. Bye-bye.